20 nations agreed to participate. 9 wanted additional MOUs on top of the standard agreements, and we executed those, and they're all available. So we signed MOUs with Australia, Ecuador, French Polynesia, Mexico, New Caledonia, Vanuatu, and the Seychelles. And you can find these on our website, sorcererexpedition.org. If countries didn't agree, we just sailed through their waters and on to the next place. Just to give you a scope of the data, because this data set is changing very rapidly. When we look at all the public databases and the scope of the data that's in there, there's about on the order of 800 genomes that have been done from the microbial world, an increasing number of draft genomes, eukaryotic genomes, including the human genome, and now there's a lot more draft ones. Just a reminder, my institute has generated somewhere between a quarter and a third of all that data set. But when you look at what we found just from sampling these filters and sequencing from them, we have quite a while ago on the order of 10 times as many genomes that could be assembled out of this information. One of the biggest surprises was every 200 miles, about 85% of the sequences were unique. In fact, we could sort of tell where in the ocean a sample came from simply by looking at the genetic code and the diversity of that we found. Some of the other findings early on were quite stunning. There was one rhodopsin-like molecule that was known for some time in the scientific literature, but you can see all the red little lines here. These are all new photoreceptors that were discovered just out of the first sample in the Sargasso Sea. This was truly a surprise. These are not associated with photosynthesis, but by light-driven biological activity. We had to develop all kinds of new tools, just trying to line up thousands of proteins takes pages and pages. At the time we first did this, we had a couple thousand. And the reasons for doing this, we can look down at the single amino acid level and try and guess the wavelength of light that activates these receptors. Because we also have the GPS coordinates of every single sequence where it derived from, we can then ask new questions about this type of data. Do we see an unusual distribution of these photoreceptors? And we're quite surprised when we did, but then it makes sense after we thought about it a little bit. Out in the Sargasso Sea, anybody who's sailed out there know it's a deep indigo blue. The organisms that survive there have receptors that see primarily blue light. You get into coastal waters where there's a lot of chlorophyll, they see primarily green light. We also have a number of new derivatives that their wavelength of activation is being determined. A study last year by a Swedish group, in fact, showed that these are light-driven organisms, not through photosynthesis, but through these visual pigments that are essentially identical in structure to what you're looking at these slides with right now. So this was totally unknown mechanisms, and probably on average, every single microbial organism in the upper parts of the ocean contain these photoreceptors. The other surprise was just how diverse the data was. And we were trying to get a graphical means to look at the data because we just had millions and millions of sequences in the computer, and trying to make sense of those was very difficult. Doug Roosh at the Institute developed this graphic recruitment tool where across the top we can put any genome sequence, for example, in an organism from the environment. And then we recruit other sequences to it based on sequence identity. And you can see we have, aside from the ones in the high 90% range, if that shows up, we have these abundant organisms that differ by as much as 15 or 20% in sequence identity. Then we have this huge group of highly related organisms. Now, this is not what was supposed to be there. All the biologists that had studied organisms in the ocean were sure there was just a few unique independent organisms. In fact, one of them that we were told we'd find in the Sargasso Sea was SAR-11. It was supposed to be a single organism. Look at this diversity, down to 55% sequence variation. Now, some people have argued that we should come up with a sequence-based definition for species. And if it's 10% or more variation, it would be a new species. Well, I don't think that appeals to us because every other mammal is within our 10% variation of us, so that would make us the same species as dogs and cats and rats. And while people understand that in Washington, they might not in other parts of the country. Here we have ones that only line up with SAR-11, but they differ by as much as 40 or 50% in their sequence variation. So what are these? We can also tell that they're largely the same genes in the same order. We can take any kind of slice of this data and make a variety of trees out of it. So these are color-coded by site, so we can compare them by site. We can compare them by whether they came out of the Atlantic or the Pacific or the Indian Ocean. The bottom one, we could ask questions about the photoreceptors, whether they see blue or green light. What we've seen is that they've switched from blue to green. It only takes a single point mutation to change the photoreceptor scene from blue to green light 4 times in recent evolutionary history. Now look what a powerful selection tool this is. It's clearly survival of the fittest. If you see blue light and you live in the Sargasso Sea, you'll survive. If you see green light, basically you won't. And it's been such a powerful selection tool, it's switched several times. You can ask any question that you want to of this type of data. Now we've added a lot more data since that set was established, and you can see the density. Each one of these little colored bars represents a single sequence discovered somewhere in the world. And these fragment recruitment plots get more and more complicated. So we have this huge degree of biodiversity that we've been trying to understand. Also, you can see areas like in the middle here where there's a lot of variation. Sometimes these turned out to be inserted viral sequences in the genome. Other places, they just look like very hypervariable regions that we're trying to understand. And you can see these in a lot of different plots. We can also assemble a finite number of these, and as we added more and more data, instead of getting intolerably complex, it starts to get clearer. So in fact, 37% of all the sequences sort of line up on 5 different taxa. So the observations that people made that there was a SAR-11 and not much else out there makes sense at some sort of primitive level when you think that all these different organisms have very similar 16S RNAs. Obviously not a good measure of diversity. So we have these limited categories with tremendous diversification within those categories. That's not what anybody would have predicted by standard evolutionary ideas. But we have this incredibly long tail where we're still having individual sequences show up that we have to sort out. Fortunately, all this technology is changing very rapidly, essentially by orders of magnitude. We had a 60,000 square foot facility up until a few months ago that because of new technology, we can shrink by over 100-fold, and the cost of sequencing is going down. So we can now do these studies very broadly. We can actually assemble genomes from organisms we've never seen that are relatively rare, and we can start to find this. So this is one of the 5 major taxa. The species, we have no idea what it looks like, but now we know what its genome looks like. But our team has also developed some extraordinary tools. We can now determine the entire genetic code of an organism from one single bacterial cell. So it's getting faster, cheaper, and much more refined. So we can take things into cell sorters, sort different populations, and sort out individual genomes. So some of the questions: how much novelty is really added by all this global ocean sampling data? How many new protein families? What's the rate of discovery?